Hi, Sylvia. I'm excited that was a great talk, and I'm excited to introduce the last talk of our oral presentation competition session. The talk is by Kyoko Yoshida, who's a postdoctoral fellow at the University of Virginia. And the title of her talk is A Multiscale Model of Pregnancy-Induced Heart Growth, Integrating Hormone Signaling and Mechanics.
Hello, can everyone hear me?
Yes.
Hello.
And
okay.
You can share your screen, make it full size.
Okay, is that good?
Yep. Great, thank you.
Okay, great. So thank you, Sylvia, for the introduction and for everyone who's here today. So today I'm going to be talking about multiscale modeling in the context of pregnancy-induced heart growth. So to start off with, what I think is really important to appreciate about pregnancy is that it really stands at this interface between mechanics and biology. So on the mechanics side, virtually all maternal organs are loaded during pregnancy or undergo growth. And so today I'm going to be talking about the heart in particular, where we see a cardiac output increase of about 45% throughout pregnancy, and then it's also associated with a systemic resistance decrease of 20% and a blood volume increase of 60%. So we see lots of hemodynamic changes occur during pregnancy. And then on the biological side, we of course see huge surges in pregnancy hormones. And so today in particular, I'm going to be talking about estrogen and progesterone. And here I'm plotting out how the circulating levels surge throughout a human pregnancy, so about tenfold over the course of 42 weeks. And in response to these changes in the mechanical and hormonal cues, the maternal heart undergoes hypertrophy or growth. So people report usually about a 20 to 30% increase in left ventricular mass that occur during a human pregnancy. And so why is this important to study? Well, it's important to study because cardiovascular conditions during pregnancy remain as a clinical dilemma. So a study in 2015 actually showed that maternal mortality is rising in the United States. And then if we look at the reasons why mothers are dying, we see that heart problems remain as the #1 leading cause of pregnancy-related deaths. And then this plot is showing reasons for these deaths. And so you can see that the rates of hemorrhage, hypertension, infection, so these rates are going down as shown in the gray. But conditions related to cardiovascular conditions as well as cardiomyopathy or heart failure, so these rates are actually rising. And so we all know that hormones and mechanics both influence heart growth. So in particular, estrogen is known to attenuate growth. So here I'm showing an experimental study in mice where we have a control mouse heart, and then we have a mouse treated with angiotensin that induces heart growth. So you can see that this heart is bigger, but once you introduce angiotensin with estrogen, you can see that heart growth is attenuated. So the size of this heart is more comparable to the control heart. On the other hand, progesterone is known to induce growth. So this was shown in an in vitro study. So this is what I'm showing the images from here. So here, when they treated the cells with progesterone, they saw that the cell mass or the cell size actually increased by about 50%. And the same group actually looked at this in vivo by treating mice with high levels of progesterone, and they saw that the left ventricular mass grew by 10%. And we can all appreciate that mechanical overload also induces growth. So because pregnancy is associated with a volume overloaded state, here I'm showing data from a volume overload study in female rats where they saw a 2-fold increase in left ventricular mass, so lots of growth. But then once they treated these female rats with volume overload plus estrogen, they saw that some of this growth was attenuated, as shown in this blue bar here. And so this is also showing us that hormones and mechanical cues do interact to influence heart growth. And so with the goal of understanding these interactions, what I'm proposing is a multiscale heart model for pregnancy. And so what this looks like is we have a mechanical model, such as the finite element model, that is able to capture the changes in mechanical loading that we see. So these would include the hemodynamic changes that I mentioned in the first slide. And this would really represent the mechanics at the system, organ, and the tissue level. So this model will give us the amount of stretch that each of the cells or tissues are feeling, which would then fed into a biological model. In this case, I'm using a cell signaling network model that will incorporate both the stretch changes as well as the changes in hormones. and then map out the intercellular pathways that lead to cell level growth. And then this cell level growth will then be fed back into the mechanical model in order to close the loop between the two models. And so today what I want to present is a simple version or a simpler version of this multiscale model as a first step. So today I'm going to be showing how we can couple a compartmental mechanical growth model to a simple version, a smaller scale cell signaling network model, and then show how we can tune this model for single inputs. So this is looking at volume overload only, estrogen only, and progesterone only. And then we'll see what happens if we try to predict a combination of volume overload plus estrogen. So to start off with, I want to go into a little bit more detail about the mechanical growth model. So what we really need here is a mathematical framework to track how the addition or subtraction of mass leads to a change in stresses and strains within the tissue. And so what you're seeing, usually when you're looking at a tissue in vivo using imaging modalities such as echo, ultrasound, or MRI, what we're observing is the tissue in its loaded state. So here on the left, we have a loaded ungrown tissue. And then on the right, we see that grown loaded tissue. So this is the observed growth that we see is the shape change between this state and this state. And so in order to solve for the stresses, what we first need to do is to unload the ventricle, and then we can tell the tissue how much to grow. And sometimes this can lead to incompatible growth. So for example, if neighboring cells or elements are growing different amounts, these meshes can overlap on top of each other in a stress-free state. But then what we do is enforce compatibility by introducing an elastic stretch. And so what this usually does is it introduces residual stresses. So that's what I'm representing here. So in the blue, we can see we have some compressive residual stresses inside the tissue, and then tensile stresses on the outside of the tissue. And then once we grow the tissue, we can reload it back to get to that final loaded state that we can observe in vivo. And so in our lab, we've implemented this growth framework within two different types of models. So first, we've used the more traditional finite element model, where you can see a realistic geometry of the left and right ventricle here. And in our models, this is coupled to a circulation model. And so these resistors and capacitors actually represent our aortas and veins, so we can capture the hemodynamics. We've also implemented this model within a compartmental model, where we actually take away the geometric complexities and then replace the left and right ventricles with a spin-wall sphere. But then we keep the circulation the same. The circulation model is the same between the two models. And what we've actually found is that both of these models do a good job of capturing volume overload or changes in growth due to mechanical perturbation. So here I'm showing our model results for predicting volume overload. So in the gray dots, we're seeing experimental data of volume overload. In the green, I'm showing how the compartmental model can match this growth very well. And in the orange, I'm showing how the finite element model can also match this trend very well. And so moving forward, I use the compartmental model to start with. And here I'm showing how we can change the model parameters within the compartmental model to match the first mechanical state that we care about, the control state. And so in the dots, I'm showing an experimental pressure volume loop from an experimental study. And then in the line, I'm showing how the compartmental model can capture that mechanics. And then what's nice about this compartmental model is that we can easily adjust it to reflect an experiment. So usually in rats, when we induce a volume overload, we introduce a fistula between the aorta and the vena cava, or a connection between the aorta and the vena cava. And so by adding this extra resistor, we can divert some of that flow. And then by adjusting the material, the model parameters, we can capture the volume overloaded state. So that's what's being shown in the red dots, the experiments, and then the solid line, the model. And so now we have the two mechanical states that we care about. And then so next I'll go into a little bit more detail about the biological model, which is the cell signaling network model. So this is a cell signaling network model for cardiomyocyte growth that was developed by Dr. Sossaman's lab here at University of Virginia. And in this model, the top line shows all of the different inputs. So you can see that things like angiotensin, which is a hormone that's associated with heart failure, is already included, as well as stretch. And what's really powerful about this model is that it integrates 14 different known pathways for heart growth. And recently, they even showed that this model is capable of predicting heart growth trends in 52 transgenic and 4 double transgenic mouse models. So that's what these red dots are showing here. So it's a very well-validated model. And so something I'm working on right now is to extend this model for pregnancy by incorporating the effects of estrogen and progesterone by collecting literature data from in vivo and in vitro studies of animals or cells that were treated with estrogen, progesterone, or both. And so as a starting point, what I've built is a smaller scale network model where a lot of the connections between the nodes actually come from this larger scale network model. Here what I'm showing is how we can recapitulate some of the data that's been shown in an in vitro study that I mentioned previously. So in this particular study, they looked at treating cells with progesterone alone, estrogen alone, or the combination of progesterone and estrogen. And in this top graph here, I'm showing how we can capture the experimental data, both at the intermediate node level. So this is looking at ERK1/2. The experimental data is shown in the solid bars here, and then the model is shown in the dash bars here. So we can capture these trends for progesterone, estrogen, or both, as well as the level, the output that we care about, which is cell area. So we can capture this increase in cell area with progesterone. They didn't see a significant increase in estrogen as what is reported in the paper, but with progesterone and estrogen alone, they actually saw a similar amount of growth as they did with progesterone alone. And so now that we have our mechanical and biological models, what I did next was to couple them together and then tune it to single inputs. So here what I'm showing are the model tuning to the volume overload data. So this is looking at what happens if we induce volume overload alone. So the different symbols here represent data from 3 different studies, and then the red line is showing how the model can fit this data. And so what I did here was to actually tune stretches that are solved for by the compartmental model, and then map them to a network model input. So the network model usually takes in a number between 0 and 1. So by changing that mapping parameter, we can match this curve very nicely, the experimental data very nicely. We can do the same for estrogen alone and then for progesterone. So here what I'm doing is mapping the relationship between the concentrations of the hormones to the network inputs from 0 to 1. And so now that we've tuned this model, what we can do next is to see what would happen if we induce volume overload with estrogen. And as I mentioned before, we expect some attenuation of growth. And so once I did this, what I observed was the curve in the blue dashed line here. So we do get this attenuated growth, which we expected to see. However, in terms of quantitative match, I've only found literature data reported for 5 days after inducing volume overload, and then 52 days. And the reason why I chose 21 days for this particular simulation was to reflect the gestation time point for rats. So in terms of quantitative match, I'll have to do a little bit more digging into the literature or actually conduct the experiments ourselves. But I wanted to take a step back and show or present how we can actually look at interactions between stretch and estrogen using this model. So what I've done here is to actually pull some of what's happening at the intermediate node level. So here I've pulled calcineurin as well as ERK1/2 and then NFAT. And what you're seeing here is that this attenuation of growth with the presence of estrogen is really being driven by the downregulation of calcineurin. That's leading to this downregulation of NFAT, as shown in this figure here, which is attenuating the decrease in cell area. And this is actually quite exciting because I've seen in other in vitro studies where they treat cells with other hypertrophic drugs or hormones, such as angiotensin or isoproternal. When they treat them with estrogen, they do see these 2 different nodes are implicated and going down as a way to regulate growth. And so I think that this is exciting because we can start building on the smaller scale network model to try to understand how these hormones should really feed into that larger scale network model. And so now to summarize, what I presented here is a first step towards building a multiscale cardiac model to capture heart growth due to volume overload alone, estrogen alone, progesterone alone, and then demonstrated how we can predict this attenuation of growth with the presence of estrogen. And so as I mentioned, what I'm currently working on is to incorporate estrogen and progesterone into this larger scale network model. But I also want to point out that I'm really interested in what would happen with the combination of volume overload plus progesterone. And this is particularly important for pregnancy because pregnancy is associated with these high levels of progesterone. And so this is a combination that has not been experimentally conducted before. So this is something I anticipate we'll have to do ourselves. And my hope for these types of modeling approaches is that we can really use these models to inform new therapeutic approaches, particularly for peripartum cardiomyopathy. So this is a very sad condition where a mother will go into heart failure towards the end of her pregnancy or after she gives birth, where lots of changes in the hemodynamics and hormones occur. So I think that this would be a really exciting modeling approach to really try to understand this disease and to come up with new therapeutic approaches. And then outside of pregnancy, I think that these models have implications for understanding gender differences in heart failure. So in particular, understanding why postmenopausal women are at higher risk for cardiovascular disease. And so with that, I'd like to thank you all for listening. I'd like to thank my lab at UVA, as well as our collaborators in Jeff Bosselman's lab, as well as our funding sources. And I'll be happy to take any questions. Thank you.